Okay, I'm going to try again. I hope to speak with you regarding whether it's possible to make a finger prick blood test for Alzheimer's disease. My affiliation is with the bioengineering department at UC San Diego. I also would like to disclose that part of this work is done from a startup company that I'm involved called Genemo Inc. Regarding my lab at UCSD, we generally work on the 4 directions. In the upper left corner, I state that we have great interest in develop molecular technologies to interrogate systematically RNA, protein, and genome interactions. We hope to review many interactions among RNA, protein, chromatin at once experimentally. And some of this work now has begun its route to be developed into potential therapeutics. On the upper right corner, we work on liquid biopsy, which we will elaborate in this talk. And apparently, liquid biopsy is one of perhaps the emerging new field related to diagnosis of disease. On the lower left corner, we also have interest to develop spatial omics technologies. We hope there's some mechanism that could potentially be reviewed. And the lower right corner, we develop machine learning and AI technologies, not only to provide statistical rigor to many of the analysis of large data set generated from the other 3 quadrants, but also we hope through these approaches, we could potentially review some principles behind all this data. Before going to the specific talk today, I would like to briefly mention about some other quadrants that I won't elaborate. On the upper left relates to molecular interactions. There is a 10-year NIH trans-NIH consortium that wishes to review many types of interactions and generally the organizational structure of the genome as well as other molecules such as proteins and RNAs inside the nucleus. So that this word nucleome relates to nucleus and ome, so everything about nucleus in 4D with 3D as the 3 dimension plus the dimension of time in total of 4D. So this consortium of 4D nucleome aims to understand the principles under nuclear organization, and apparently the technologies relates to genome interaction and RNA genome, protein genome interactions among the most focused developments of the 4D nucleome consortium. Relates to what we've been trying to do on the upper left corner. We hope to develop technologies that would systematically review, for example, all RNA interactions that would include perhaps microRNA and messenger RNA interactions that we probably are more familiar with, as well as other potential interactions between messenger RNAs, between pseudogene RNAs and messenger RNAs. And a couple of years ago, we have released this Mario mapping RNA interactions in vivo tool to achieve that. Also, that now people have started to recognize that RNA can associate with the genome. So the attachment of RNA on the genome now is regarded as potentially another layer of the epigenome. RNA can modulate gene expression by interacting with the chromatin. So in the recent 5 or 6 years, now that there is a cohort of recent technologies to systematically review which RNA can attach to which part of the genome, that leads to discovery of many new chromatin-associated RNA and start to review their functions. So MARGI, mapping RNA genome interaction, is one of the recent technologies, and now seems to be widely adopted and leveraged and to interrogate this type of interactions. And on the lower left corner, so protein-protein interaction is apparently important type of molecular interactions that underline essentially perhaps every aspect of protein function. And we all know how challenging it is to experimentally review novel protein-protein interaction. It can take weeks, if not months, to achieve that for discover one interaction. And so we wanted to provide molecular technologies that would have experimentally review hundreds and thousands of protein-protein interactions from human cells at once. So the proper-seq technique, which I will not develop, I will not dive in depth today, is a way to barcode many diverse proteins by unique DNA, and then convert the question of interrogating protein-protein interaction into DNA sequencing as a readout to read out a protein-protein interaction. So these tools are proper-seq for protein-protein interaction, MARGI for RNA-DNA interaction, Mario for RNA interaction are published technologies. And we have an ongoing piece of work that hopefully would allow us to review many RNA binding proteins de novo, as well as their RNA targets. So we would like to call it PRINSIC, protein RNA interaction by sequencing. We hope that can be released on bioRxiv. And with all of this, we hope to integrate all this data to hopefully provide a leap to our understanding of these types of molecular interactions. I also would like to mention about endeavor by many labs at UC San Diego to initiate this UC San Diego Liquid Biopsy Research Center. And so I've worked with our faculty teams at UCSD, ADRC, Alzheimer Research Center, and as well as UCI, Alzheimer Research Center, to develop the Alzheimer disease liquid biopsy analysis focus here at the Liquid Biopsy Research Center. We also have a strong interest and research under the direction of breast cancer in reproduction, especially assisted reproduction for in human clinic, as well as immunotherapy. We have a growing interest in leveraging single cell and also circulating RNA and DNA to interrogate the potential of help to immunotherapy. And so with all this said, let me converge to one question, not necessary to provide an answer, but provide some discussions to see whether there is a possibility for disease diagnosis using peripheral blood. Before discussing about the current research, I also wanted to mention about some of the lessons learned from the recent 10 years, including the cautions and opportunities that are laid out. I would like to do a case study using this company called Theranos as an example. So Theranos was a startup company, but very quickly grew into a giant biotech. At year 2015, not too long ago, it was evaluated as a $10 billion US dollar company. That makes it one of the rhinoceros in the biotech field. And what Theranos does is to develop blood tests that require only very small amount of blood for the test. And the founder was Elizabeth Holmes. She founded Theranos in 2003. And she founded the company partially by dropping out from her college. She was a college student at Stanford University, and she dropped out from Stanford at her age of 19 in order to found Theranos. And one of the reasons that she felt that she could make an impact was because she discovered a pain. When we say a pain in the startup field, we refer to the need, the desperate need from the society. And the desperate need is as follows. A simple blood test would require a punch and a venous blood draw of usually 10 ml of blood. You may have experienced this. Sometimes it's not enough. 2 or 3 draws are required. For some of people who have to do this once in several days, once a week, for months, this could be extremely painful. And I have close relatives who have to do this once a week for several months. And after some time, and there was so much scar on your elbow, and the nurse couldn't even puncture into the vein anymore. You can see that how painful a patient would be to try to get numerous additional punctures and without any success. That would even make great stress to the nurse. So this was a pain to the society, and this was literally a pain to any person who have to experience this. So what Elizabeth felt is that if we can change this to a finger prick, then the pain could be greatly reduced. It means if she could only use a tiny amount of blood to revamp how the blood tests are done. And also the market was tremendous. So there are some numbers cited here by MarketWatch.com, and the liquid biopsy market itself very soon is expected to be at $6 billion annually. So that was sufficient to support many new ventures, including what she believed she could do. The turning point for Theranos was very quick, and I wanted to point out that even though in 2015, the company raised $700 million, it's tremendously successful. 3 years later, the company went down to bankruptcy. The evaluation was 0. Part of the reason was that it wasn't successful in deliver blood tests that would only require very little amount of blood. And the company claimed that it had those technologies. It was discovered that the company didn't, that very quickly the funds were exhausted and the company was closed in 2018. I feel that the knowledge learned partly is that there is need. The demonstration of the need was obvious. I recall that when our lab moved from Illinois to California, one of the things here in California was that in the local pharmacy like Walgreens, we walk into there, we see Theranos machines for blood, for finger prick. You can literally go to your local pharmacy and put your finger there, boom, puncture, and then you should get your result in a few days. So that says how popular it could be if you have that technology. You could change how blood test practice is done. However, whether the technology is improbable, impossible at all, now knowing the failure of that exercise, I wanted to review this whole thing with you together by thinking about what molecules in the blood that potentially can be used for diagnosis purpose. Perhaps everything about the blood has been tried, including pulse, pressure in traditional medicine, and circulating DNA, any cell in the blood, a white blood cell, any kind of white blood cell, any metabolite, metabolites apparent important, sugar level, peptide, any circulating proteins. We have people here mentioned exosomes. And one of the molecules that was perhaps much less considered was extracellular RNA. And this is a class of molecules that were poorly tested, poorly investigated, and we wanted to ask if it's possible to measure RNA. And I wanted to give you a few reasons. One of the reasons was that many of the other types of molecular species cells have been heavily pursued already. And at least 3 years ago, those molecules have not advanced into a finger prick level of blood for test. And for extracellular RNA, I wanted to give a very brief introduction. So these are RNA existent outside of the cells, even until the beginning of the year 2000, this type of molecule was not discovered. People didn't know it exists, partly because that very few RNA existed outside of the cell for a good reason. So in the human blood, for example, the extracellular space is filled with RNAs. We need RNAs. People need RNAs because any extracellular RNA most likely would be a virus. So in our blood, outside of the cell, there are so many RNAs that are trying to clean up and digest RNA as much as possible as a defense system for humans. Nevertheless, starting in around the year 2000 and more reviewed more recently, that there are tiny amount of RNA outside of the cell in the human blood in circulation. These RNA tend to be small, tend to be protected. So either because perhaps these are already after RNA's digestion, those are remaining small fragments that somehow not completely digested, or they are protected by exosome or by some other ribonuclear protein. So there are such a species over there. And several years ago, we contributed to quantify in human blood the concentration. We measured it to be approximately 1 nanogram per ml. So that concentration compared to the typical concentration of cellular RNA, this is probably 1,000-fold less. Roughly speaking, the concentration of extracellular RNA in the human blood is 1 over 1,000 of the concentration of cellular RNA. And these extracellular RNA typically are smaller than full-length messenger RNA. So we measured it to be most likely between 20 to 150 nucleotides in length. Now that there are many other papers out there that the quantified size and concentration, I believe the range is probably generally accepted now. The bottom line is these molecular species do exist. And the question that presented itself to ourselves was that how to effectively measure extracellular RNA from blood, especially from blood serum, and whether that such measurement can be used for diagnosis purpose at all. By saying diagnosis purpose, of course, we mean people, diagnosing people. At that time, we spoke with clinician friends, clinician colleagues, physician colleagues, and we asked what it takes to test another biomarker, a potential biomarker in patient cohorts. We got a very consensus response. That is to say that, well, if you need 10 ml of blood for measurement, you need to take that blood draw from people, and then you need to run a trial. It takes several years. In some of the examples I will show, one of them took 6 years, the other one took 15 years. And you need to draw those samples, and you wouldn't be able to validate any of your new biomarker in the next several years. We said, well, are there already ongoing trials we can participate? They say it's probably unlikely. While there are many ongoing trials, many of them work with blood. However, those blood had their designated measurement. Say they wanted to measure certain protein as a potential biomarker, they need to use that blood. However, if your new test only requires a tiny amount, say microliter level, microliter meaning a drop of blood. That variation is smaller than the variation of the nurse in every blood draw, right? When they draw a whole tube, whether it's several microliter more or less, that is the error zone. That is totally fine. If most of the trials, they have the ability to spare microliter level, tiny amount of blood for another measurement, if the other measurement only require a very tiny amount of blood to measure. Knowing that, Genemo, our startup, in collaboration with UCSD, we started to try to develop a technique that potentially only require 1 drop of blood for measuring, for sequencing extracellular RNA. And I wanted to mention 2 of our elite collaborators, Dr. Shu Chen here at UCSD and Dr. Irene Su here at UCSD Morris Cancer Center, that we collaborated in this paper to demonstrate from breast cancer recurrence patients how literally from 1 drop of blood, you can have some way to distinguish, to predict which of the chemotherapy treated people would potentially develop recurrence of cancer. And let me go back to the principle. So we wanted to try, and we thought it's possible, and it's not completely impossible, because we knew at that time the concentration of extracellular RNA is 1,000-fold smaller than the cellular RNA. But still, at around the year 2015, it was already 6 years after the initial publication of the first publication of single-cell RNA sequencing. So at that time, single-cell RNA sequencing was already becoming a reliable technology. We knew, people know that we can reliably sequence RNA from a single cell. The total amount of RNA from a single cell is about the total amount of 1 drop of blood serum. The reason is that the volume of 1 drop of blood serum, say 30 microliter, that is about 1,000-fold larger than the volume of a cell. Now, the total amount of RNA in 1 drop of blood, in theory, is comparable in the order of magnitude to 1 drop to 1 cell. So from a back of envelope calculation, George Zhou, who I will mention in a minute, at Genemo, thought that there's not a physical barrier between the difference. If there's a similar amount of RNA there, possibly there could be a technology. Having that said, it still took us several years to optimize that, to make it an actual technology. So we delivered this technology to the community, which we call small input liquid volume extracellular RNA sequencing, SilverSeq in short. So what SilverSeq does is that it only requires the input of 5 microliter of serum. That is only like 1/7 of a drop of blood. So here is an example that I wanted to demonstrate and to show the quality of RNA sequencing library. From 1 drop of blood serum, so we could split it into 8 aliquots. Here I didn't split it into the same volume. We had the first 2 aliquots of 3 microliter and 2 aliquots of 5 microliter, 6 microliter, and 7 microliter per se. And here are the size distribution of the final sequencing library as a quality check to see if we could generate a good size distribution that is suitable for a typical Illumina sequencing platform. And I wanted to very quickly to jump to illustration. So this clinical.gov, clinicaltrial.gov registered trial run by Dr. Irene Su. It's called a POISE trial, short for Predictors of Ovarian Insufficiency in Young Breast Cancer Patients, where she followed chemotherapy-treated young breast cancer patients for 6 years. And among that 5 to 6 year follow-up, some people had recurrence of cancer. Very unfortunately, some didn't have recurrence, and still pending unsolved clinical challenge is there any molecular biomarker that potentially can help to predict who may have recurrence? That apparently has a huge impact to how to manage the patients, how to potentially watch and treat them. And in this particular follow-up, there were 28 blood samples from people that are from people with recurrence in this window of follow-up. There were 68 samples from women who didn't have recurrence in this 5-year follow-up. And we were able to measure extracellular RNA by sequencing using 1 drop of blood, literally speaking, for the same reason as I mentioned before. And most of the blood were used for the original purpose of the original trial. In this extracellular RNA profile, turned out that we could use cross-validation to demonstrate that you could actually have a fairly accurate prediction of who would have recurrence of breast cancer through their blood. So I wanted to quickly summarize. So extracellular RNAs are different from intracellular RNA in size and concentration. Now, SilverSeq technology can sequence extracellular RNA, and extracellular RNA sequencing can be used to classify the blood serum samples from patients with and without cancer recurrence. I mentioned that this work was led by 2 scientists at that time at Genemo, Josh Zhou, and at that time, a visiting PhD student to my lab and intern at Genemo, Dr. Qiuyang Wu. Now that Qiuyang is Dr. Qiuyang Wu, by those 2 young scientists. So they worked together around the year 2017, 2018, 2019. They published their work jointly as co-first authors in 2019. And that was 3 years ago. 3 years later, in March of 2022, and I was fortunate to join their wedding here at San Diego with 2 of our lead authors at the center, and also 2 of our other co-authors, now Dr. Yuan Liu and Dr. Zhi Jieqi here for a science bounded couple. And with that, I wanted to get into Alzheimer's. 1 in 9 people who are of age 65 or older are living with Alzheimer's disease. Unfortunately, 2/3 of these people who live with Alzheimer's disease are women. In 2060, by the time that the audience here become elderly, it is projected that 14 million people will be living with Alzheimer's disease. How many of those people is? It is twice of the total population of the Washington state. You imagine that there's 2 Washington states' entire population of people will be living with Alzheimer's disease by the time that you are at the age of elderly. A very brief overview of some of the symptoms and pathology of this unfortunate disease. People with Alzheimer's start to be confused with where they are and what time it is. They start to forget what year it is, whether they are in US or in China. They don't know who's the president. They start to demonstrate poor judgment. You see some of the people start to speak publicly for things that no one should speak in that way. And they start to be forgetful. They forget where they just came from. They have trouble in repeating simple sentences who were just told. And in A very brief pathology. Neuron death is vastly observed. Brain size shrink, and this called beta amyloid plaque start to form. You can actually visualize them in human brain. Unfortunately, treatment methods are extremely limited. You may have seen recent news. There was one newly approved drug in 20 years. Unfortunately, that newly approved drug didn't demonstrate good response either even after it's approved. So for more than 20 years now, there haven't been a substantial development on the treatment front. That probably contributes to why the projected number of Alzheimer's people rise so quickly, because there's essentially no way to effectively treat them. Then what should we do? I would like to cite from Alzheimer's Foundation, Alzheimer's Association, of is there any potential way to consider how to potentially make a change? I would like to quote, "What if we could diagnose Alzheimer's disease before symptoms started? The hope is future treatments could then target the disease in its earliest stages before irreversible brain damage or mental decline has occurred." So that has become the main thought of what to do scientifically from here on. How to detect it early? Brain is a very special tissue. Tissue biopsy, while popular perhaps for most other tissues, is nearly impossible for brain because the damage is obvious. You literally cannot take a piece of brain unless this is horribly devastating. Usually, if this is not a brain tumor, it is very rare to do tissue biopsy. Without tissue biopsy, you do not have cellular or molecular measurements of the organ or the tissue that is potentially have a problem. And so that speaks to how diagnosis is very limited. You pretty much are limited to the symptoms rather than having an objective measurement. So almost the only thing that left to try is blood. So the question becomes obvious. Is it possible to develop a blood test for Alzheimer's? And to us, it's whether it's possible to develop the test based on extracellular RNA. Other molecules are being tried at this moment where we particularly would have wanted to know whether the extracellular RNA represent any of the gene expression of the brain. If so, is that a venue or a bridge that we can leverage to learn something about the progression of the disease from the brain? And there we were fortunate enough, partly because we can use very little blood to work with at that time ongoing trial at UC San Diego led by Dr. Edward Kuhl. So this was a 15-year follow-up study of people who were recruited from year 2000 to year 2015. And the people were recruited to the study when they were of age 65 or older. And when they were recruited, they were clinically diagnosed. If they didn't behave or have any Alzheimer's symptoms, they are classified as controls. And if they were already diagnosed when they entered the study, they are classified as AD in the next follow rows. And there were a group of people when they originally were enrolled, they were diagnosed as normal. But during the follow-up, at some time after several years, the diagnosis changed, right? They became older. So they started to have either dementia or diagnosis into Alzheimer's disease. So mild cognitive dementia is usually an earlier diagnosis with a smaller, less symptoms, but they could further develop into Alzheimer's. And so we have this group of people who were normal to begin with, but as time goes by, they change into disease diagnosis. And so here we identified 35 participants. We identified by that after their deaths, their brain pathology, this is analysis of the actual brain, could actually confirm whether these people are normal or Alzheimer's disease. So their diagnoses are confirmed by pathology. And throughout their enrollment, their blood were collected at the particular time. So the graph on the right shows that every row is a person, and every dot is a blood collection time. So you can see that some of the people, the blood was collected multiple times, and in total, there were 164 blood plasma samples were collected. And before we interrogate about the sequenced extracellular RNA in this blood, we also wanted to know what happens to people's brain. In fact, that in the past 10 years, there were several large cohorts trying to generate gene expression profiles from Alzheimer's brain and compare to control brain. Here, I would like to highlight 3 cohorts that are done by Mayo Clinic, Mount Sinai, and Rosemap. I named those 3 because those 3 are NIA, National Institute of Aging-funded cohorts. And in these cohorts, they used RNA sequencing on several brain regions that are related to the Alzheimer pathology in approximately 80 to 100 plus donors with or without AD. And they carried out RNA sequencing. And it's actually new, even before those cohorts, there were very few genes at gene expression level. By gene expression level, I mean RNA abundancy or protein abundancy level, not post-translational modification, but just about abundancy of the RNA or the protein level. Very few genes are consistently up or down regulated in AD. And this was actually confirmed when we analyzed these 3 cohorts together. From the 3 cohorts, we wanted to identify a subset of genes that are consistently upregulated or become higher expressed in AD. So we had our own requirement. This is not the only possible way you can analyze this data, but with our requirement, we require certain fold change and statistical significance to call a gene to be higher in AD than normal, and then we need it to be showing up in at least 5 of the brain regions in at least 2 cohorts. Altogether, there were only 28 genes that satisfied this criteria. Also, that confirms the idea that not many genes can be reproducibly shown to expressed at higher level in AD versus control brain. And we wanted to start from those 28 genes to see how do these genes behave in the blood? How do their extracellular RNA behave in the blood? In the left, I would like to show all the genes with respect to their extracellular RNA level between AD donors and control donors in our cohort. So the lowest distribution of all genes show t-test with positive being extracellular RNA of this gene is higher in AD than control in our cohort. And what we see is in general that there is perhaps not a clear bias to higher or lower. Some genes express higher and lower that conforms to what would you expect from a distribution. I wanted to bring you to the top level. The top level are the genes with expression increase in AD brains, meaning the 28 we identified in the previous slide. Those are the genes that in previous cohorts to show some consistency in upregulation in AD. Now, if we plot them into with our blood serum, blood plasma extracellular RNA level, you see that most of them are still centered at 0, but a small population shows up to be high in AD. Perhaps suggests that a subset of these consistently upregulated genes in brain have some consistent upregulation in their extracellular RNA level in blood. I wanted to show this again on the right panel. On the right panel, every column is one of the 28 genes that were defined as consistently upregulated in AD brain. And on the y-axis is a comparison between AD and the control blood. So a positive value means that this gene not only exhibited consistent upregulation in brain, but also in our data shows higher level in extracellular space in blood. And what we see is that more of the genes are higher in blood than smaller amount of genes are lower in AD in blood, suggesting that there's still some consistency between what you see in blood and what you see in brain, which is unusual because otherwise we don't really know whether the blood reflects anything of the brain. And among these, apparently the gene on the right would be a potential candidate because in blood, again, it shows a good separation between AD and control. So I wanted to expand that for you and to see how it behaves. So if we expand this gene, and on this panel C, this is a plot of their this gene PHGDH expression level in every cohort and every analyzed brain region that related to Alzheimer's disease. So the box plot in red are the distribution in AD brain, whereas the box plot in blue is the distribution in the control brain. You see that this gene is consistently upregulated in AD in nearly every cohort analyzed and every AD-related brain region analyzed. These were from 3 previous cohorts. And in our cohort here in panel D, you see that the extracellular RNA level is consistently higher in blood than control. That says that potentially PHGDH may be a biomarker. I wanted to quickly summarize here. PHGDH is consistently upregulated in AD brains in 3 cohorts, and PHGDH genes extracellular RNA is higher in AD patients' blood than control. So the almost obvious immediate question is what cell types in the brain express PHGDH, and how does PHGDH expression change in each cell type? Does the PHGDH protein level increase in AD hippocampus in brain? Hippocampus Haimati is potentially the most critical brain region to the disease. Does the protein change? How does the protein change in hippocampal, human hippocampus in AD would be a critical next question to potentially start to understand why there may be a consistent gene expression that now we observed in multiple cohorts. I wanted to start with the first question: what cell types in brain that express this gene? And with this question, we reanalyzed a single-cell RNA sequencing dataset released relatively recently in 2019 that generated a single-cell RNA sequencing from 24, 15, and 9 people with no AD, early AD, and late AD. So here we are replotting the expression level of PHGDH in 7 cell types in human brain. The reason we can get cell type information is because now we have single cell expression data, and using that single cell expression data, we can have assessment of what cell type it is. So here on the columns, we have astrocytes, endothelial cell, excitatory neuron, inhibitory neuron, microglia, oligodendrocyte precursors. Those represent perhaps the main cell type you would expect in the brain. And we have 3 columns here in 3 colors that represent the single cells from people with no AD, those are control, people with early stages of Alzheimer and late stages of Alzheimer in green and in blue and red respectively. And you see this is the single cell expression level of PHDH. What we see here is that PHDH is expressed in astrocyte, oligodendrocyte precursor, not in every cell type. In astrocyte per se, you see that from no to early to late Alzheimer's, there is a stepwise progression of the expression level. So now we started to see what cell types express this gene, and also the change of expression level of this gene actually starts from the early stage of the disease. And I promised to go into human hippocampus. So here at UCSD, we were able to carry out a study of a 21 postmortem brain. We immunostained this protein in their hippocampuses, including 10 control and 11 AD brains. So here is an example. On the right, you see immunostaining of this protein in the AD brain in hippocampus. You see this staining of the protein that appears to be obvious as composed to the staining of the PHDH protein in the control brain that doesn't seem to stain as clearly. So if we ask a pathologist's opinion about what cells are stained, so these cells you can see that seem to have a star shape are mostly astrocytes. So those are supporting cells to work with the neuron, and they seem to express a high level of the protein, especially when the AD get progressed. You can quantify this. So these start to demonstrate that something you measure from different patient cohorts, even from the blood, actually indeed reflect some changes from the brain and indeed relate to the correct brain location and correct cell type in the diseased brain. And finally, there is an open challenge in the field is while this person is alive, right? That's most important is before you get the postmortem brain. Is there any molecule that you can measure that potentially correlate to this person's cognitive ability? Meaning, do you have any objective measurement that can help to diagnose about the person's cognitive ability? And cognitive ability currently is measured by you essentially have a standardized test, like you ask the person to carry out some tasks, to answer some questions, repeat some sentences, and then that's quantified, right? And so one of those quantification is using a task that is called a dementia rating scale and eventually give a quantitative score. The larger the score it is, by answering those questions, it means that clinically it's interpreted as a better cognition. A person with dementia or Alzheimer's disease usually would reflect it as having a lower score. Now, at least for those people that we have got the brain, we can quantify their PHDH protein level in their hippocampus. It turns out that these two measurements have a strong correlation. It's a negative correlation. The higher the molecular level of PHDH it is in the person's hippocampus, the poorer the person's cognition ability it is. Despite that, I want to acknowledge that this is so far a small size study. Only a small number of people who had this test in our clinic, and we had their postmortem brain. But still, now that we seem to have an indication, there is a possibility of a connection between a molecular measurement and a person's behavior, a person's cognitive ability. And so with that, I want to summarize PHGDH mRNA exhibits a sequential increase in brain astrocytes as AD progresses. PHGDH protein level is high in Alzheimer's hippocampus, and hippocampal PHGDH level negatively correlates with the person's cognitive ability. I wanted to mention that this is led by my colleague, Assistant Professor Dr. Xu Chen here at UC San Diego, and Riccardo Canandrelli, a scientist in my lab, to make these observations. That's the very last. I wanted to go back to the question: can plasma PHGDH extracellular RNA level predict AD before clinic diagnosis? After all, that was our hope. Is there any way to diagnose it early? Well, the person apparently is alive. You can't get to the brain. Can you have a quantitative way to identify the disease even before the symptoms develop? In fact, there is a group of converters whose data we have not used at all, right? We mentioned that in our 15-year follow-up, there were people who were normal, however, unfortunately converted to AD during the follow-up. We didn't interrogate this set of data at all, right, so far. So if I were to plot their PHGH extracellular RNA level one by one, now in every panel is a person. It's converter 1, converter 2, and so on. If I were to plot their plasma PHGH extracellular RNA level on the y-axis and their follow-up year on the x-axis, then we have everyone's data in one panel. So we reordered time zero, year zero as a year their earliest clinical diagnosis of the disease was found. So before they were diagnosed as Alzheimer's were the years that now we label them as -1, -2, -3, and so on. After the first diagnosis into a disease, the years are labeled now as positive years. This way, so we recentered the first time of diagnosis as year 0, so that it's more comparable. And from this data, you can see that the PHGDH extracellular RNA increase as the person progress into clinically recognized diagnosis. So we can see this in this summary plot. So every dot in this summary plot is the slope of the fitted curve across time. So now that we have reduced the entire collection of longitudinal data of a person of this 15-year follow-up into one number, that number is a slope. The higher the slope, the larger the increase it is across the follow-up. So the first group in blue are the slopes of our control. The green, the last group, are the slopes of the converters. You see that there is a populational difference. So in controls, the slopes are almost near 0. Most of them are near 0, meaning that while these people are old and become older, as long as they do not develop into Alzheimer's pathology, the PHGDH level in their blood seems stable. However, if they convert into AD, there is usually a spike of increase of their PHGDH level in their blood. So I wanted to quickly summarize: PHGDH extracellular RNA level increases before AD symptoms develop, and this data nominate PHGDH extracellular RNA as a biomarker for early diagnosis of AD. So this work was led by Dr. Zhangming Yan, at that time a postdoc, now a lead scientist in Novo Nordisk. With that, I wanted to quickly mention some aftermath. So there are patient advocate groups who are very much eager to learn about any new potentials in any development on either diagnosis or therapeutic fronts. So being patient is one of those venues. They heavily reached out and to us and mentioned about the possibility of early diagnosis may allow for early treatment. What does PHGDH do? What is the potential mechanism? Why it could serve as a biomarker? So the known function of this phosphoglycerate dehydrogenase PHGDH gene is an enzyme. This is an enzyme essential for serine synthesis, especially for the brain. So in the glucose metabolism pathway, glucose can be converted into the following two molecules. Either it could be converted into pyruvate, or it could be converted through a branching route into serine and then glycine. So PHGDH is actually a controlling enzyme to the route from glucose to serine. So with PHGDH, you could brain could produce serine and glycine in the brain, which are essential nutrients. So if we were to review about this known mechanism of PHGDH, how does this known mechanism connect to the current knowledge of potential causes of AD? As one of the highly hypothesized cause of AD is so-called excitotoxicity. That is firing of neuron all the time, and it's essentially makes the neuron too tired and exhausted, and finally causing neuronal death. So the neuron has become excited all the time and essentially become toxic, excitotoxicity. That requires the calcium to be released from presynaptic to postsynaptic neuron through for the excitation of the postsynaptic neuron. The calcium goes into a channel. So the membrane channel, one of the perhaps most important membrane channel is so-called NMDA channel, receptor channel. This channel would be closed most of the time, but when it opens, the calcium comes in. It opens when two ligands are bound. One is glutamate, shown in red. The other is serine or glycine, shown in blue. So you see that this is like a gate with two keys. You require the two keys to be both turned open to open the gate. And one of the two keys is synthesized by PHGDH. So we hypothesize that if you have a high level of PHGDH in the elderly's brain, you make this control system too often prone to open, because previously you need to satisfy two criteria. You know that glutamate is often abundant, especially in glutamate neurons. Now that you make the other key more likely to be at the open state, that potentially potentiates excitation all the time, that leads to excitotoxicity. So with that, I wanted to mention a part of our team, including, I mentioned Dr. Edward Koo, Xu Chen. I would like to acknowledge our pathology professor, Annie Henniker, Douglas Glasgow on the clinic front, and many of our other collaborators and many of the team members I mentioned in the talk, but not all of them. I hope to acknowledge every of them. And finally, I wish to mention an anecdote. So if we Google serine, this essential amino acid, we would find many ads of this supplement. In fact, for me, even before I Google, perhaps AI was already intelligent enough to already feed me serum and ask, are you going to buy serum? Probably because I analyzed serum-related research, and somehow AI believed that I wanted to buy this supplement. So this supplement is touted to be a brain booster, to boost your brain. Let me first say, if I wanted to argue this supplement has any use, what would I say? If it has any use, I feel that this probably has some benefit to the children with a mutation of PHGH gene. The people with a mutation of PHGH gene actually would develop a familial disease. So this is sometimes called a PHGH deficiency or serum deficiency, because PHGH is essential for making serum, and serum is an essential nutrient for brain development. You can imagine that if there's a mutation in this gene, the young brain would not have sufficient serum and it would not develop fully. So that people with mutations of this gene often develop this phenotype called a microcephaly, and that means small brain. That is very unfortunate. And you can imagine that the children with microcephaly, because that they do not have sufficient serum, and a serum supplement could be potentially helpful to these children. Nevertheless, by now you probably feel that these ads are not targeting this small population, right? It's really wanted to sell, perhaps want to sell to a large population, including like boost your brain. Most of the time, these are sold to adults. Would serum supplement help adults? Turn out that most cohorts failed to confirm any decrease in serum levels in the brain or cerebral fluid from adults. So serum levels were not decreased in AD. And our study also confirmed that PHGDH didn't decrease, but increased in AD's patients' brain and blood. So therefore, this supplement may not be as helpful as it may claim. And you may know that this supplement has its own challenges that, for example, we mentioned a candidate mechanism for it to overactivate neuron activity and essentially potentially could lead to excitotoxicity, but serum also has other effects to lead to progression of cancer. And so our study got very quickly took up by professional societies and neurologists. So there are, for example, ASMB quickly warned about this supplement, and the KPBS public radio station in its evening news also made this news.
Researchers at UC San Diego have identified a gene marker for Alzheimer's disease. KPBS Science and Technology reporter Thomas Fudge says this means we can better predict Alzheimer's and maybe find the path to a cure.
The study looks back on 15 years of blood testing and observation of hundreds of people over 65. And researchers say the result is an undeniable connection between an overactive gene and the development of Alzheimer's disease. UCSD bioengineering professor Sheng Zhang says their findings were a surprise. That's because the gene and serine, the nutrient it produces, play an important part in brain development.
It's like an overshoot of the good guy, of the good stuff that seems to mark unfortunate disease outcome.
The study tracked the development of Alzheimer's over time in people who were symptomatic and asymptomatic. The expression of the overactive gene, that goes by the acronym PHGDH, shows up as these dark star-shaped cells, a strong contrast to the same cells in a healthy brain. So far, Zhang says they have proven a correlation between the active gene that produces serine and Alzheimer's, and while they haven't proven cause and effect, they think that's what's happening.
Our hypothesis is that a pronounced oversupply of serum makes neurons too active for too long a time and thus become toxic,
which can lead to the conditions that lead to Alzheimer's. There is no cure for Alzheimer's disease, and the UCSD researchers have not discovered one. But Zhang says there are ongoing experiments on mice and on brain samples that could lead to a way to tone down the overactive gene and the overproduction of serine,
to either ameliorate the development of the disease or even reverse the development of the disease. Those are the tests that we are currently doing.
Ironically, some businesses sell serine as a dietary supplement to increase brain function. Based on what he's learned, Zhang says for those who may suffer Alzheimer's, those supplements may be just what they don't need. Thomas Fudge, KPBS News.
So the take-home message I got from this KPBS Evening News is that while the team members do all the work, the PI gets onto the TV. The other good note is that so this ad, Boost Your Brain, previously was on Google and Amazon. So now that somehow it's been taken off. So it looks like that there's the society as a whole, the community as a whole may have taken some advantage of the warning that we were able to provide through our research. So with that, I would like to thank everyone here with me. Also, I want to open for questions. I already see many questions